हेलो दोस्तों, वेलकम टू ग्रासरूट्स एकेडमी। और आज हम एक टॉपिक के बारे में बात करने वाले हैं जो है कि केल्विन साइकिल। अब देखिए, केल्विन साइकिल सबने ही पढ़ा हुआ है। अब आप एक टीचर से क्या एक्सपेक्ट करते हो कि वही रिएक्शंस, वही पीजीए, वही 1,3 बाय पीजीए, वही पीजीएएल, वही रुबिस्को, इस शायद सब कुछ आपने पढ़ा हुआ है। आप 10th से भी पढ़ रहे हो, 12th में पढ़ा है, आपने बीएससी में पढ़ा है, एमएससी में पढ़ा है। इसमें नया क्या है? इतने वीडियोस पढ़े हैं यूट्यूब में, मेरा भी एक वीडियो ऐड हो जाएगा वहाँ पे, फिर वही रिएक्शंस, वही रटना। लेकिन आज का लेक्चर स्टार्ट करने से पहले शायद हम कुछ नया करने की कोशिश करेंगे। विल गिव यू अ न्यू पर्सपेक्टिव ऑन दी C3 साइकिल। शायद ये दूसरों से काफ़ी कुछ अलग होगा। वो चीज़ें आज मैं आपको बताऊंगा जो शायद आपको नहीं पता है C3 साइकिल के बारे में। तो C3 साइकिल स्टार्ट करने से पहले हमें यह देखना है कि कुछ C4 भी हमने सुना हुआ है, कुछ कैम भी हमने सुना हुआ है। तो हमें यह पता है कि कुछ प्लांट्स में C3 साइकिल होती है, कुछ प्लांट्स में हम कहते हैं C4 प्लांट्स होते हैं जिनमें C4 साइकिल होती है, जिसको हम हैच एंड स्लैग पाथवे भी कहते हैं। और उसी तरफ एक और प्लांट ग्रुप्स हैं जिनको हम कहते हैं कैम प्लांट्स, जिसमें कैम साइकिल या कैम मैकेनिज्म पाया जाता है। अब सारे ये CO2 फिक्स करते हैं। हमें पता है कि C3 ग्रुप्स में वी नो व्हाट इज C3 पाथवे। C3 पाथवे बेसिकली कैप्चर्स एंड फिक्सेस CO2। देन व्हाट इज C4, व्हाट इज कैम। तो ये जो C4 है, ये जो कैम प्लांट्स हैं, ये C3 से कैसे डिफरेंट है? क्योंकि देखिए, हम पढ़ते तो अपने स्कूल्स और कॉलेजेस में C3 भी हैं, हम अपने स्कूल्स और कॉलेजेस में C4 प्लांट्स भी पढ़ते हैं, हम अपने स्कूल्स और कॉलेजेस में कैम प्लांट्स भी पढ़ते हैं। ये हमने सारे ही पढ़े हैं वहाँ पे। अब इनमें डिफरेंसेस क्या हैं, क्यों हम पढ़ते हैं? क्या C3 पाथवे जो है वो C4 से डिफरेंट है? क्या कैम पाथवे जो है वो C4 से डिफरेंट है? क्या ये आपस में डिफरेंट है? तो दैट मीन्स कुछ तो इनमें फर्क होगा कि हम C4 और कैम को पढ़ रहे हैं। अब लेट मी टेल यू, ये जो C4 पाथवे है, C4 पाथवे का काम क्या है भाई? C3 पाथवे क्या करता है? लेट्स टॉक अबाउट C3 पाथवे। सी व्हेन वी टॉक अबाउट C3 पाथवे, वी नो कि C3 पाथवे बेसिकली कैप्चर्स एंड फिक्सेस द एटमॉस्फेरिक CO2। भाई एटमॉस्फेरिक CO2 को कैप्चर करता है, उसको फिक्स करता है, एंड इट इज कन्वर्टेड टू ऑर्गेनिक फॉर्म। It is converted to organic molecules in the organic form, so we get organic molecule. We know, जैसे आपको बहुत लोगों को पता होगा, ग्लूकोज बनता है, फ्रक्टोज बनता है। What are they? They are basically organic molecules. And ultimately, then what is C4? क्या C4 is also involved in the fixation of this atmospheric CO2? क्या this also forms the organic molecules? और क्या सेम हमारे कैम प्लांट्स में भी यही हो रहा है कि this is also a pathway which basically fixes the atmospheric CO2 and converts it into organic molecules? तो ये बहुत कंफ्यूजन रहती है। Now, अब इस कंफ्यूजन को दूर करने के लिए सबसे पहले मैं आपको ये बताना चाहूँगा कि all the plants, हमारे बायोस्फीयर में, हमारे अर्थ में जितने भी प्लांट्स हैं, they will all have C3 pathway। तो ये तो बड़ा मतलब मेरे ख्याल से कंट्राडिक्टरी स्टेटमेंट हो जाएगा, क्योंकि आप कहोगे सर C4 प्लांट्स में भी क्या C3 पाथवे होता है? क्या कैम प्लांट्स में भी C3 पाथवे होता है? हाँ, बिल्कुल होता है। यही तो मैं आपसे कह रहा हूँ कि ऑल दी प्लांट्स, whether they are C3, whether they are C4, whether they are CAM, they will all have, without any exception, they all have what is the C3 pathway. They all have the C3 cycle. They all have the C3 pathway. Look at this. That means ये जो C4 प्लांट्स हैं उनमें भी C3 है, C4 प्लांट्स में भी देखो C3 है यहाँ पे, हमारे कैम प्लांट्स हैं यहाँ पे भी C3 है, और C3 में तो हमें ऑलरेडी पता है कि C3 पाथवे है ही। तो क्वेश्चन ये है कि जब सारे ही प्लांट्स में C3 पाथवे है तो फिर C4 का क्या काम? फिर ये कैम प्लांट्स का काम ही क्या है भाई? क्यों हम पढ़ रहे हैं? क्यों फालतू हमें कॉलेजेस और स्कूल्स में बर्डन किया जा रहा है कि भाई कैम पाथवे पढ़ो, C4 पाथवे पढ़ो, जब सभी प्लांट्स में C3 है? That means, and we know C3 is the pathway which basically fixes the atmospheric CO2 and converts it into organic molecules. तो यहाँ पे भी ऐसे ही हो रहा है, यहाँ पे भी ऐसे ही हो रहा है। फिर ये C4 क्या है? फिर ये कैम क्या है? हम क्यों पढ़ते हैं सारा कुछ? तो शायद ये हमारा एक पहलू होगा जिसके बारे में आज हम थोड़ा सा डिस्कशन करेंगे, ठीक है? लेकिन सबसे पहले चलिए बात करते हैं C3 की। लेट अस स्टार्ट विद दी C3 प्लांट्स। सी द क्वेश्चन इज, when we start with these C3 plants, when we start with these C3 plants, we know, so when we start with the C3 plants, we know ये जो C3 cycle है, which is also called as the Kelvin cycle. Sometimes we call it Kelvin-Melvin cycle. Actually this cycle was discovered by a scientist whose name is Kelvin Melvin. ठीक है? And it was during the World War II that the C14 isotope of carbon was made available. ये जो C14 isotope है, which is the radioactive isotope of the carbon, it was made available during the World War II. So these guys, that is Melvin and Kelvin, they used this C14 isotope of carbon to discover a set of reactions in the plants. And they said that these reactions which were discovered by using C4 isotope, they call these reactions as the Kelvin cycle. And they said that the end product of these reactions are the triose phosphates. That means what is the end product? The end product of this Kelvin cycle is basically what is a triose phosphate. Is this very clear? So this is what is the Kelvin cycle. This is what they discovered. They discovered the reactions which ultimately led to the production of the triose phosphates. And these reactions, this discovery was so important that in the year 1961, this Kelvin, Mr. Melvin Kelvin, he was awarded a Nobel Prize. So that means this Kelvin cycle is actually very important. It has even got a Nobel Prize in it. Now the question is, see, when we talk about the C3 pathway, when we are talking about this C3 cycle, let me tell you, the C3 cycle, as I have told you, this is found in all the plants. चाहे वो trees हों किसी तरीके के, चाहे वो shrubs हों, चाहे वो grasses हों, चाहे herbs हों, चाहे वो हमारे algae हों, चाहे bryophytes हों, चाहे pteridophytes हों. Is this clear? They all have what is the C3 pathway. That means I can say the C3 pathway is the fundamental pathway through which the atmospheric CO2 enters into the plants and it is converted into the organic molecules. That means it is the fundamental pathway. This is the only pathway through which the atmospheric carbon or the CO2 can enter into the different plants and it is being converted into the organic form. That is the biomolecules. That means it is the C3 cycle which will fix the atmospheric carbon, convert it into carbohydrates, your fats, your proteins. We know they all have a backbone of carbon. And how do we get this carbon? We get this carbon through what is the C3 cycle. So that means this is the fundamental pathway. So this you have to remember. Sabhi plants mein we have the C3 pathway. Now the question is, when we talk about C3 plants, अब आपके मन में क्या आता है भाई, कौन से प्लांट्स ऐसे होते हैं C3 प्लांट्स डेली यूज में, क्या आपको पता है? Can you tell me certain plants which are using or their products which you use daily? आप C3 प्लांट्स को डेली यूज करते हो, शायद आपको नहीं पता होगा. आप C4 प्लांट्स को भी यूज करते हो, आप कैम प्लांट्स को भी आजकल गर्मियों में यूज कर रहे हो. Can you tell me certain important C3 plants which are part of your daily life? Yes. भाई आप सबसे ज्यादा क्या खाते हो? हमें पता है, if you go to South India, if you go to North East, their staple diet is rice. क्या है? चावल खा रहे हो भाई, राइस खा रहे हो. What is rice? Rice we know is a C3 plant. राइस इज अ C3 प्लांट. अब हम जो नॉर्थ इंडियंस हैं, नॉर्थ इंडियंस क्या खाते हैं यहाँ पे भाई? रोटी खाते हैं, चपाती खाते हैं यहाँ पे. तो रोटी किससे आती है? रोटी बनती है व्हीट से. व्हाट इज व्हीट? गेहूं क्या है? व्हीट क्या है? दैट इज अ C3 प्लांट. और जो लोग आप पीजी में रह रहे हो, पीजी में आपको पता है सबसे ज्यादा सब्जियां क्या खाते हैं आप? आलू. और बहुत लोगों की इनफैक्ट आलू इज अ फेवरेट डाइट. अब मैकडी में जाओ, वहां पे यू ईट फ्रेंच फ्राइज, इट इज ऑल व्हाट? पोटैटो. इनफैक्ट यू नो आयरलैंड में द स्टेपल डाइट ऑफ पीपल इज बेसिकली व्हाट इज पोटैटो. तो हमारी डाइट में या तो हम गेहूं ज़्यादा खाते हैं, या हम चावल ज़्यादा खाते हैं, या हम पोटैटो लेते हैं. व्हाट इज पोटैटो? पोटैटो इज ऑल्सो अ C3 प्लांट. सो दैट मीन्स वन ऑफ द मेजर staple diet that we consume, it is coming from the C3 plants. And on the other side, if you talk about the C4, अगर हम C4 की बात करें तो भाई कौन से ऐसे C4 प्लांट्स हैं जो कि हमारी डेली लाइफ में इंपॉर्टेंट है? ऐसे कौन से C4 प्लांट्स हो गए? चीनी खाते हो, शुगर के बिना तो कुछ भी नहीं है. ना आप आइसक्रीम खा सकते हो, ना चाय पी सकते हो, ना कॉफी का कोई स्वाद है. तो व्हाट इज शुगर केन? शुगर केन इज बेसिकली नथिंग एल्स, इट इज अ C4 प्लांट. इट इज अ C4 प्लांट. आजकल आप मार्केट में जाते हो, वहां पे आप कॉर्न खाते हो, कॉर्न, स्वीट कॉर्न. ठीक है, तो व्हाट इज कॉर्न? ये जो भुट्टा खाते हैं हम, जो मेज है, मेज, कॉर्न, स्वीट कॉर्न, वन एंड द सेम थिंग. व्हाट इज स्वीट कॉर्न? इट इज नथिंग एल्स. जो मेज है, इट इज बेसिकली व्हाट इज अ C4 प्लांट. तो look at this group. Sometimes they ask you this. That means you have to match. So we know now some important C3 plants that you can identify with. You know certain important C4 plants you can identify. For example, we have sugarcane, we have maize, we have sorghum, we have millets. List is never ending. But I am talking about plants which you can identify with. ठीक है? Similarly, हमारे पास कैम भी है. अब भाई कैम प्लांट्स कौन से हो गए जो हमारे डेली यूज में अक्सर हम यूज करते हैं? जैसे हमने देखा है बच्चों को पाइनएप्पल बहुत पसंद है. ठीक है, बड़े होते जाते हो आप पाइनएप्पल कम खाते हो. लेकिन जो बच्चे होते हैं उनको पाइनएप्पल बहुत पसंद होता है। तो अल्टीमेटली व्हाट इज पाइनएप्पल? वी नो पाइनएप्पल इज अ कैम प्लांट। और दूसरा, आजकल गर्मियां हैं, देखो अब गर्मियों में आपके मम्मी पापा को आइसक्रीम पसंद है, कौन सा फ्लेवर खाते हैं? वेनिला। भाई आपको तो बटर स्कॉच ज्यादा पसंद है, आपको स्ट्रॉबेरी पसंद है, लेकिन जो मम्मी पापा जो बूढ़े लोग होते हैं उनको सिंपल फ्लेवर्स पसंद होते हैं, जैसे वेनिला। व्हाट इज वेनिला? यू नो वेनिला इज बेसिकली नेम ऑफ एन ऑर्किड। वेनिला इज बेसिकली नेम ऑफ एन ऑर्किड। ये जो वेनिला का फ्लेवर है, दिस वी आर गेटिंग फ्रॉम ऑर्किड्स which are nothing else, which are monocots, which belong to Orchidaceae family. So vanilla is an orchid which belongs to Orchidaceae family, and we know vanilla is nothing else, it is a cam plant. Vanilla is a cam plant. We know cactus. What is cactus? Cactus is basically what is a very common cam plant. So look at these common plants, you know, that you can identify with. भाई लिस्ट तो बहुत है, बहुत सारी फैमिलीज है, वो तो हम बाद में बात करेंगे ही, टू कंफ्यूज यू। अभी मेरा काम आपको कंफ्यूज करना नहीं है, जस्ट टू मेक थिंग्स मोर इंटरेस्टिंग दैट यू कैन आइडेंटिफाई थिंग्स विद, यू नो, ठीक है। सो नाउ वी नो सर्टेन C3 plants, we know certain C4 plants, we know certain CAM plants that we have. Now since we are talking about the C3, we are talking about the C3 pathway. Now the question is, see, when we talk about the C3 pathway, we know ये जो C3 pathway है मतलब क्या? सेट ऑफ रिएक्शंस, जो आपने 12th क्लास में बहुत अच्छे से रट्टा लगाई थी, सेट ऑफ रिएक्शंस। तो रिएक्शंस तो आप रट्टा लगा लोगे, लेकिन व्हाट अबाउट द अंडरस्टैंडिंग, व्हाट अबाउट द एनालिसिस, जहां से क्वेश्चन आ रहा है। कोई रिएक्शंस में तो आपको कोई एग्जाम में लिखने को बोलेगा नहीं कि देयर इज एन एग्जामिनर और यू आर गोइंग फॉर सम मे बी एंट्रेंस एंड यू हैव टू राइट द एंटायर सेट ऑफ रिएक्शंस। नो, दैट इज नॉट रिक्वायर्ड. What is important now is the analysis. So when we talk about the C3 plants or C3 cycle, we know all the enzymes of the C3 cycle, they are basically confined to that is chloroplast. मतलब हमारा जो C3 साइकिल है, ये पूरा जो C3 साइकिल है, ये कहाँ पे है? अगर मैं कहूँ कि ये क्लोरोप्लास्ट है हमारा, दिस इज द क्लोरोप्लास्ट, वी नो दिस इज द ग्रेना, these are the granas over here, we know granas are interconnected with each other. So now the question is, if you look at this, this is the stroma, ये हमारा स्ट्रोमा है यहाँ पे, which is nothing else, stroma is nothing else, it is aqueous solution. जैसे माइटोकॉन्ड्रिया में मैट्रिक्स है, हमारा साइटोप्लाज्म है सेल के अंदर, सिमिलरली क्लोरोप्लास्ट के अंदर क्या होता है? वी हैव स्ट्रोमा, इट इज नथिंग एल्स, इट इज बेसिकली aqueous solution, you know, and ultimately here we have all the enzymes of the C3 pathway. Look at this, all the enzymes of the C3 pathway are basically present in the stroma of the chloroplast. Is this very clear? So now we know the location also, that means it is found in the stroma of the chloroplast. Now when we talk about the C3 cycle, हमने अपनी आसानी के लिए अब किताबों में तो भाई बहुत सारी चीजें दे रखी हैं, लेकिन हमने अपनी आसानी के लिए क्या किया कि वी हैव डिवाइडेड दीज रिएक्शंस इनटू 3 पार्ट्स. वी डिवाइडेड दीज रिएक्शंस इनटू 3 पार्ट्स. जैसे हमने कहा कार्बोक्सिलेशन, हमने कहा रिडक्शन रिएक्शंस, हमने कहा रीजनरेशन रिएक्शंस. ये हमने अपनी आसानी के लिए, फॉर आवर ओन ईज, वी हैव डिवाइडेड, वी हैव कैटेगराइज दीज रिएक्शंस. अदरवाइज रिएक्शंस तो वही हो रही हैं, A to B to C to D, लेकिन हमने अपनी आसानी के लिए इनको 3 पार्ट्स में डिवाइड कर दिया. जैसे कार्बोक्सिलेशन रिएक्शंस, we have the reduction reactions, and we have the regeneration reactions. So this is very clear. Now the question is, ये देखो, see this. Now I say this is a leaf. ये हमारी पत्ती है, यहाँ पे देखो भाई, सब कुछ तो फोटोसिंथेसिस कहाँ होता है आपको पता है? फोटोसिंथेसिस will take place inside the leaf. Photosynthesis takes place inside the leaf. अब अगर हम लीफ की बात करें तो लीफ में यहाँ पे एपिडर्मिस भी है. This is the upper epidermis. We know this is the lower epidermis over here. We know there's a stomata that we have. Look at this. यहाँ पे हमारा स्टोमेटा भी है यहाँ पे. Is this very clear? And then below the epidermis we have the mesophyll cells. यहाँ पे देखो, ये हमारे mesophyll tissue है, ये हमारा mesophyll tissue है यहाँ पे. And we know this mesophyll tissue is divided into palisade cells. ये हमारे palisade cells हैं और ये हमारा spongy tissue है. This is the spongy tissue that we have. This is the palisade cells that we have. We all know this. And together we'll call it what is mesophyll. We call it mesophyll. This is the mesophyll tissue that we have. Now the question is, most of the chloroplast, जहाँ पे photosynthesis हो रहा है, most of this chloroplast is present inside the, ये देखो, ये हमारे जो palisade cells हैं. तो palisade cells are the ones where the main photosynthesis will take place. This is over here. You can see this is the chloroplast. ये हमारा क्या है? This is the chloroplast present in the mesophyll cells. Ultimately what happens? Ultimately CO2 तो हमारे atmosphere में, ये देखो CO2, and we know during the daytime the stomata is open. We know the stomatas are open over here. And what happens when the stomata is open during the daytime? We're talking about the daytime. What happens? The CO2 enters inside the leaf. The CO2 will enter inside this, CO2 comes inside, and when it comes inside through the stomata, it will enter through the membrane, cell wall or membrane ko cross karegi, it enters the chloroplast, and as it enters the chloroplast, the CO2 reaches the stroma. मतलब क्लोरोप्लास्ट के थ्रू, मतलब ये जो हमारा स्टोमेटा के थ्रू द CO2 enters and finally it enters the cell, finally it enters the chloroplast and it reaches the stroma. So that means now if we talk about this chloroplast, अगर हम ये chloroplast की बात करें यहाँ पे, we know this is the chloroplast roughly, ये हमारी दो membranes हैं यहाँ पे, ये हमारा grana है यहाँ पे, so that means ये जो हमारी CO2 है, CO2 has come inside the स्ट्रोमा. दिस इज ऑल स्ट्रोमा. CO2 अंदर आ गई. एंड वी नो हमारी जो मेम्ब्रेन्स होती हैं, चाहे हम प्लाज्मा मेम्ब्रेन की बात करें, चाहे हम क्लोरोप्लास्ट मेम्ब्रेन की बात करें, चाहे हम सेल वॉल की बात करें, दे आर मोस्टली pervious to all the gases, you know. That means they are permeable to all the gases. Oxygen, CO2, these are the important gases, and we know gases can easily pass through the membrane. चाहे हम NO की बात करें, nitric oxide, we know is a very important signaling molecule. This can also easily enter through the membranes. We don't need any proteins for their transport. That means through the membranes the CO2 can enter, through the membranes the O2 can also enter, there is no assistance which is required, finally it reaches what is the stroma. अब भाई आपके घर में एक मेहमान आ गया, अब CO2 आपके घर में आ गया, तो आप क्या करें? That means we have to capture this. भाई कोई मेहमान आपके घर में आएगा तो आप जाओगे उससे प्रणाम करोगे, you will say hello and hi, तो ultimately that means somebody has to be there in the stroma to receive this CO2. And we know who receives this CO2. We know it is a 5 carbon compound. That means हमारे स्ट्रोमा में we already have a 5 carbon compound. Look at this. So there's a 5 carbon compound which reaches out and which accepts this CO2, which will welcome this CO2. And what do we call this 5 carbon compound? We know this 5 carbon compound is called as RUBP. सबको पता है भाई, what is the name of this 5 carbon compound? We know this 5 carbon compound is called as RUBP. That is ribulose 1,5 bisphosphate or biphosphate. This is ribulose 1,5 biphosphate or bisphosphate. This is a 5 carbon compound which is the acceptor molecule for this inorganic CO2 which is coming from the atmosphere. Now both of them, this RUBP will combine with the CO2 and we get a 6 carbon compound. We'll get a 6 carbon compound. But it's not that easy. It's not that easy. Look at this. Look at this reaction over here. So we have the ribulose 1,5 biphosphate, which is a 5 carbon compound. Look at this, 1, 2, 3, 4, 5. So this is RUBP roughly. Ribulose 1,5, so that means there's a phosphate here also in the first position. We have a phosphate in the fifth position also. This is very clear. And at the second position of this RUBP, we have a double bond. This is O. This is what is carbonyl carbon. So this is the molecule of RUBP, and at the second position, which is more important, we have this double bond O. This is the carbonyl carbon. अब ये जो हमारी CO2 आ रही है, look at this CO2 will come and this CO2 will actually interact with the second carbon, which is the carbonyl carbon of this RUBP. And now we will get over here, what is a 6 carbon compound? We get a 6 carbon compound. And we all know this, that the 6 carbon compound is highly unstable. And it will only exist for a very transient, for a very short period of time. And till date, we have not been able to isolate this 6 carbon compound. It is highly unstable. Is this very clear? And ultimately what happens, ये जो 6 carbon compound है, this 6 carbon compound would be hydrolyzed. That means we also require water. So this 6 carbon compound is ultimately hydrolyzed and we get what? We get 2 molecules of 3 carbon compound. We get 2 molecules of 3 carbon compounds. Look at these reactions over here. But the question is, who catalyzes these reactions? Who is the enzyme which will catalyze these reactions? And we know the enzyme which catalyzes all these reactions, this is, you all know this is the Rubisco. What is the enzyme? This is the Rubisco. Look at this. So, who captures the CO2? Who fixes the CO2? It is Rubisco. Where it is present? It is present in the stroma. And what is this reaction? This is the first reaction that we are talking about, which are the carboxylation reaction. So this is the carboxylation reaction, which is the first reaction. After this, we know we have reduction reaction. After that, we have regeneration. We have divided the C3 into 3 parts. The entire C3 cycle divided into 3 parts: carboxylation, reduction, and regeneration. So right now, we are focusing on the first set. That is the first part, which is the carboxylation. And who catalyzes this? We know it is the Rubisco. And ultimately, it captures the CO2, it fixes the CO2, attaches the CO2 to RUBP. We get a 6-carbon intermediate which is highly unstable, and this is hydrolyzed. It is hydrolyzed to 2 molecules of 3 carbon. So this is very clear. And what do we call this 3-carbon compound as? You all know, you are all smart enough. We call this 3-carbon compound as 3-PGA. This is 3-phosphoglyceric acid. What is this? We know this is nothing else. This is 3-PGA. This is 3-PGA. That is, this is 3-phosphoglyceric acid. Now the question is, we are talking about these reactions over here. And in these reactions, let us focus something on the Rubisco. Because Rubisco is a very important enzyme because it catalyzes the carboxylation reaction. Now if we talk about Rubisco, we know Rubisco, which is present in the leaves, this is one of the most abundant proteins which is found in the leaf cell. So most abundant soluble protein which is found in the leaf cell is basically what is Rubisco. And according to an analysis, 20 से 25 से 30%, from 20 to 30% of the soluble protein which is found in the leaf is nothing else, it is basically what is Rubisco. So देखिए कितना इंपॉर्टेंट है, ठीक है. तो लीफ में सबसे ज़्यादा मात्रा में जो प्रोटीन पाया जाता है वो Rubisco है. Not only this, we know the most abundant protein on the earth. अगर मैं अर्थ की बात करूं तो अर्थ में सबसे ज़्यादा जो abundant प्रोटीन है, सबसे ज़्यादा जो मात्रा में पाया जाता है, that is what is Rubisco. And according to an analysis, the amount of Rubisco present on this earth is approximately 40 million tons. 40 million. 1 million is 10 lakhs. So 40 million tons of Rubisco is present on this earth. That means this is one of the most abundant proteins which is found on this earth. That is what is Rubisco. अब इस Rubisco की अगर हम बात करें तो we know Rubisco is a 16 subunit protein. There are 16 subunits which are present in the Rubisco. तो Rubisco में हमारे 16 subunits हैं, and out of the 16 subunits we know 8 subunits are synthesized or they are encoded in the nucleus, and other 8 subunits they are encoded by the chloroplast genome. So 16 subunits में से जो 8 subunits हैं they are encoded by the nuclear genome, and the rest 8 subunits are encoded by the chloroplast genome. क्योंकि हमें पता है कि chloroplast is semi-autonomous and the chloroplast has its own DNA which has the genes for the subunits of Rubisco. So this should be very, very clear. That means for the synthesis of Rubisco, the nucleus and the chloroplast genome, they have to come together. They have to contribute. Both have to contribute. And the genes of Rubisco which are present in the nucleus, the genes of this Rubisco which are present in the nucleus, we call them as RBCS gene. क्या कहते हैं इनको? RBCS genes. और जो genes of the Rubisco which are present in the chloroplast, we call them as RBCL genes. Look at this, RBCL genes. ये कहाँ पे है? These are found in the chloroplast. और ये वाली genes कहाँ है? RBCS genes, they are found in the nucleus. Look at this. So RBCS genes, RBCL genes, S का मतलब small subunits, L का मतलब large subunits. That means I told you out of the 16 subunits, जो 8 subunits which are encoded by the nuclear genome, these are small. These are the small 8 subunits. और ये जो 8 subunits which are encoded by the chloroplast genome, which is RBCL, these are the large subunits. So this is very clear. That means 8 large subunits from the nucleus, from the chloroplast, and 8 small subunits, that is from the nucleus, they will come together, and through chaperones, which are special enzymes, these units, they will amalgamate together, they'll come together, and we get a single large enzyme, which is what is Rubisco. Now one point to ponder over here, one point to highlight over here is that these RBCS genes, look at these RBCS genes, which are encoding the small subunits and which are found in the nucleus. These RBCS genes, for the expression, they require light. That means for the expression of these genes, we need basically what is? The light is needed. So that means if there's no light, there would be no Rubisco. So this should be very, very clear. And we call these C3 reactions as dark reactions. We know in our colleges, in our schools, we have been taught this, that photosynthesis is divided into 2 parts, the light reaction and the dark reactions. And what is a dark reaction? Dark reaction is nothing else. It is basically C3 cycle or the Kelvin cycle. So the question is, why do we call the Kelvin cycle as dark reactions. Do they take place during the night or what? क्या ये C3 cycle हमारी रात में हो रही है या अंधेरे में हो रही है या इसको लाइट नहीं चाहिए? That is why we call it the dark reactions? No. Let me be very clear that C3 cycle will only take place in the presence of light. अगर लाइट नहीं है तो C3 cycle नहीं होगी. अभी आपने एक एग्जांपल देखा कि one of the most important enzymes of the C3 cycle, which is Rubisco, the expression, the synthesis of Rubisco itself requires light. तो फिर ये डार्क रिएक्शंस कैसे हो सकती हैं? दैट मीन्स इट इज साइंटिफिकली इनकरेक्ट, इट इज अ मिसनोमर, इट इज साइंटिफिकली इनकरेक्ट टू कॉल द C3 cycle as dark reactions. The C3 cycle requires light. It will only take place in the presence of light. As we move forward, we'll focus on this, we'll throw more light on this, कि why C3 cycle requires light. So this is the first example that I've given you over Rubisco, कि synthesis of Rubisco also requires light, but there are more points which we'll talk about later. Now the question is, see, we have talked about this Rubisco. We know about the Rubisco now. Is this clear? So 16 subunits. This is one of the most abundant protein present on this earth. Now one more characteristic feature, one more peculiarity about this Rubisco is that this Rubisco, if you look at the name of the Rubisco, we call it ribulose 1,5 biphosphate carboxylase oblique oxygenase. तो Rubisco का दूसरा नाम क्या है? We call it ribulose 1,5 biphosphate carboxylase oblique oxygenase. That means ये जो हमारा Rubisco है, if you look at this enzyme Rubisco, this is functioning as a carboxylase also, and it functions as oxygenase also. Now why the hell is this? That sometimes we call this enzyme as carboxylase, sometimes we call this enzyme as oxygenase. So again it creates a confusion over here. That means a single enzyme can have two different kinds of catalytic activities, carboxylase as well as oxygenase. Is it true? Yes, this is true. Because if you look at this enzyme Rubisco, this Rubisco was evolving at a time when the oxygen was making its presence in the atmosphere, and that is why this oxygen and CO2 structurally they are very similar molecules, and they started competing for binding to the active site of Rubisco. So that means ये जो two different activities, two different catalytic activities, carboxylase and oxygenase, this is nothing else, this is an evolutionary load on the enzyme. This is evolutionary load. This is how the enzyme has evolved. We cannot do away with these activities. So the question is, under which condition the enzyme will function as oxygenase, and under what conditions the enzyme will function as carboxylase? Now this is a lot of confusion again. कि photosynthesis, when we talk about photosynthesis, what we are presuming, हम ये assume कर रहे हैं कि photosynthesis में जो enzyme है, this will always function as carboxylase, and this we don't discuss. ये तो हम डिस्कस ही नहीं करते, तो सिर्फ हमें नाम ही पता है बस कि कार्बोक्सिलेज ऑब्लिक ऑक्सीजनेस, लेकिन इसकी रेलिवेंस क्या है? डज दिस एंजाइम एक्चुअली फंक्शन एज ऑक्सीजनेस? एंड इफ इट फंक्शंस एज ऑक्सीजनेस, व्हाट इज द इंपॉर्टेंस ऑफ दिस? इज दिस वेरी क्लियर? सो व्हाट इफ दिस एंजाइम फंक्शंस एज ऑक्सीजनेस, विल इट फिक्स CO2? Can it fix CO2? No, it cannot fix CO2. This enzyme will only fix CO2, it will only catalyze the carboxylation reaction if this enzyme functions as carboxylase. Now the question is, when the enzyme will function as carboxylase and when the enzyme will function as oxygenase? How does the enzyme know this? Because ultimately these activities will ultimately determine the photosynthesis. कितना photosynthesis हो रहा है? What is the quantum of photosynthesis? This is all determined by the activity of the Rubisco, and you know it can be very detrimental also. So the question is, see, when we talk about this Rubisco, we know this Rubisco enzyme has more affinity towards CO2. This is very clear. This Rubisco enzyme is found to have more affinity. It has in fact inherent activity. The inherent activity of the enzyme, it has more affinity towards CO2. इसकी तरफ इसकी affinity ज़्यादा है. Whereas it has less affinity. This enzyme has found to have less affinity towards oxygen. It has more affinity towards CO2. फिर तो कोई प्रॉब्लम ही नहीं होनी चाहिए, क्योंकि enzyme has more affinity towards CO2, फिर तो it will always fix CO2, it will always recognize CO2 because it likes CO2, it will capture CO2. Then what is the problem? Yes, there's a problem. I say कि enzyme has more affinity towards CO2, it has less affinity towards oxygen. Look at the concentration of these two gases. We know the concentration of two gases. If you look at the CO2, the amount of the CO2 present in the atmosphere is not even 0.1. हमारे एटमॉस्फेयर में 1% तो छोड़ो, 0.1% भी CO2 नहीं है. What is the percentage of CO2? This is 0.034%. Look at the percentage. It's not even 0.1. Kitni kam CO2, and still we say this is a greenhouse gas. It is so detrimental. It causes global warming. Look at the percentage. It's not even 1%. It's not even 0.1%. On the other side, how much is the amount of oxygen? We know the percentage of oxygen present in the atmosphere. This is 21% approximately. So even though this enzyme Rubisco has more affinity towards CO2, but look at the percentage of CO2. It's not even 0.1. Even though it has less affinity towards oxygen, but look at the percentage of oxygen, this is 21%. That means there's a high probability that this Rubisco will keep interacting with oxygen, even though it has less affinity, but because the percentage of oxygen is more in the atmosphere, so the probability of interaction of this Rubisco with the oxygen increases multiple times. भाई जब CO2 है ही नहीं, तो interaction कैसे होगा CO2 के साथ? The interaction would naturally decrease. And this is a problem, This is a problem. That the ratio of CO2 and O2 in the atmosphere itself is a problem. And that is why, my friends, for every 2 to 3 carboxylation reactions, मैं क्या कह रहा हूं, हर 2 से 3 carboxylation reactions which are catalyzed by Rubisco, one oxygenation reaction would also be catalyzed. तो हर 2 से 3 carboxylation reactions के बाद एक oxygenation reaction भी होगी, क्योंकि percentage of oxygen is very high in the atmosphere. And so the question arises कि whenever this enzyme functions as carboxylase, भाई acceptor molecule कौन है? Acceptor molecule तो वही है ना, 5 carbon. This is RUBP, ribulose 1,5 biphosphate. This is the acceptor molecule. Now this is Rubisco. This is the Rubisco. Now if this Rubisco functions as carboxylase, look at this. If this Rubisco functions as carboxylase, this will capture CO2. And it captures CO2. This will give rise to the same reaction that we saw earlier. It will give rise to a 6-carbon compound, which is highly unstable, which is existing for a very transient period of time. This will break down into 2 molecules of 3-carbon compound. And we know this 3-carbon compound is nothing else. It is 3-PGA. This is 3-phosphoglyceric acid. So this is the normal Calvin cycle reaction. This is the reaction which we call as the carboxylation reaction. This is the reaction. We call this as the carboxylation reaction. This is what we have seen, the first reaction of the Calvin cycle. Now the problem is, when this enzyme functions as oxygenase, let us see this. The problem arises when the enzyme functions as oxygenase. Look at this. Again the 5-carbon compound. Look at the 5-carbon compound. This is what is RUBP, ribulose 1,5 biphosphate. This is the enzyme Rubisco that we have. Rubisco, now this enzyme functions as oxygenase. This enzyme functions as oxygenase. Now the question is, when this enzyme functions as oxygenase, this will not capture CO2. That means now it captures what? Now this will capture oxygen. So it interacts with oxygen. So what do we get here? Do we get a 6-carbon compound? No, we will not get a 6-carbon compound. What we get over here is a 5-carbon compound. So we will get a 5-carbon compound instead of a 6-carbon compound. So what do we do with this 5-carbon compound now? अब इसका क्या करें हम? Can we break it into 2 molecules of 3-PGA? We cannot break it into 2 molecules of 3-PGA. That means this 5-carbon compound now would be further hydrolyzed. This would be further hydrolyzed, and we get 1 molecule of 2-carbon compound, and we'll get 1 molecule of 3-carbon compound. Whereas when this enzyme functions as carboxylase, we will get 2 molecules of 3-carbon. But when this enzyme functions as oxygenase, we are not getting 2 molecules of 3-carbon. We are getting 1 molecule of 3-carbon compound and 1 molecule of 2-carbon compound. This 3-carbon compound remains the same. This is the 3-phosphoglyceric acid. This will enter the C3 cycle. As usual, this enters the C3 cycle. This is consumed by the C3 cycle. But the problem arises with the 2-carbon compound. What do we do with the 2-carbon compound? And what is this 2-carbon compound called? We call this 2-carbon compound. Look at this 2-carbon compound. We call this 2-carbon compound as phosphoglycolate. What is the name of this 2-carbon compound? We call this as phosphoglycolate. So, what is the problem? What is the problem here? Yes, there is a problem. The problem is, ये जो plant है, this plant cannot use this 2-carbon compound. This 2-carbon compound phosphoglycolate, it cannot enter the C3 cycle. It cannot be used by the C3 cycle. and it starts appearing as a waste. That means whenever this enzyme functions as oxygenase, the 2-carbon compound, which is phosphoglycolate, cannot be further utilized by the plant. This cannot be utilized by the plant, and this will start appearing as a waste. This will start appearing as a waste. So how do we use this? Because in producing this compound, we have spent a lot of energy. We have already spent a lot of energy, It takes energy. We know the cost of one carbon to fix one CO2 in the plant, this is 3 ATP and 2 NADPH. So if we waste this 2-carbon compound, that means actually we are wasting ATP and NADPH. So what do we do with this? And on the other side, if this starts accumulating, because it is a waste, and if it starts accumulating, if this compound starts accumulating, we know it will tend to inhibit one important enzyme of C3 cycle. This will inhibit an important enzyme of C3 cycle, which is called as triose phosphate isomerase. तो C3 cycle में एक important enzyme है which is called as triose phosphate isomerase. Look at this. This is an important enzyme of C3 cycle. It converts 3-phosphoglyceraldehyde to DHAP. This is the enzyme. So if the concentration of this 2-carbon compound phosphoglycolate increases, we know it tends to inhibit this enzyme. This compound ultimately will inhibit this enzyme, that is triose phosphate isomerase, which is important component of C3 cycle, and that is the C3 cycle will stop. It would be inhibited. Now this is a problem, that whenever this Rubisco functions as oxygenase, it produces phosphoglycolate accumulates because it cannot be used by the plant, and if it starts accumulating, it becomes very toxic. If it starts accumulating, we know it becomes highly toxic, and it can inhibit an important enzyme of the C3 cycle, which is triose phosphate isomerase. That means the C3 cycle itself will stop. Look at this. So now you can relate how dangerous is this oxygenase activity of Rubisco. That means it can even kill the plant. That means it can even kill the plant. So what do we do with this? The question is, how do we take care of this oxygenase activity of Rubisco? So look at these plants. Look at this. Look at these plants over here. Look at this phenomena over here. Now see, the question arises when we are looking at the C3 plants. When we are looking at the C3 plants, we have seen the first reaction, which is the carboxylation reaction. And we'll talk about all this aspect later. But coming back to the C3 cycle, let us not lose the track. So when we come back to the C3 cycle, we have already seen the first set of reactions. The first set of reactions we have already seen, which are called as the carboxylation reactions. The second set of reactions we know. What is that? The second set of reactions we know are the reduction reactions. We have the reduction reaction, and in the reduction reaction what is happening? Look at this. The name itself is very clear. In the reduction reaction, by carboxylation reaction के बाद क्या बना था? We had two molecules of 3-PGA, 3-phosphoglyceric acid. This is what we got at the end of the carboxylation reaction. So what is happening in the reduction reaction? In the reduction reaction, this 3-phosphoglyceric acid, we know this is an acid. This is being converted to aldehyde. And this is what is reduction reaction. Very simple. That means during reduction reactions, what happens? This molecule of 3-PGA, this is 3-phosphoglyceric acid. This is first converted to an intermediate, which is 1,3-bi-PGA, which is 1,3-bi-phosphoglyceric acid. And in the next step, we know this 1,3-bi-PGA. We know this 1,3-bi-PGA is converted to 3-phosphoglyceraldehyde. So we get a molecule of aldehyde. That means from acid, ultimately, we get a aldehyde. And we know conversion of acid to an aldehyde, this is a reduction process. We know this process over here, this is a reduction reaction. This reaction requires a lot of energy. The reduction of acid into aldehyde requires a lot of energy. For conversion of 3-PGA to 1,3-bi-PGA, we know we need a molecule of ATP. ATP is required, it is converted to ADP. In the next step over here, we need a source of electron, and the source of electron here is NADPH. This is NADPH, which is the source of electron. So look at these reactions. These reduction reactions require a lot of energy in the form of ATP, in the form of electrons. Finally, we get what is aldehyde. So this is the relevance of the reduction reactions. Now, the question is, if you've gone through mitochondria, if you've gone through mitochondrial respiration, that is the glycolysis, these intermediates are also found in glycolysis also. वहाँ पे भी यही हो रहा है, वहाँ पे we start with 3-PGAL, then we have 1,3-bi-PGA, then we have 3-PGA, then we have 2-PGA, then we have PEP, phosphanol pyruvate, which will finally give us pyruvate. That means these reactions are also found in mitochondrial respiration, which is part of glycolysis. और यहाँ पे भी ये reactions हो रही हैं, लेकिन यहाँ पे these reactions are inverted reactions. Inverted, उल्टी हो गई यहाँ पे कहाँ से start हो रहा है? PGA से. Mitochondria में कहाँ से start होता है? PGAL to 1,3-bi-PGA to 3-PGA. And that is why sometimes these reduction reactions are also called as what? These reduction reactions are also called as, that is, mitochondrial reversal reactions. They are also called as the glycolytic reversal reactions. तो हम इनको क्या बोलेंगे? हम इनको बोलेंगे glycolytic reversal reactions. Reversal reactions. Is this very clear? The only difference is even the enzymes are same. The only difference is that in the case of chloroplast, we use this coenzyme which is NADPH, whereas in the case of mitochondria, we do not use this NADPH, we use NADH. Otherwise, the reactions are same, but they are inverted over here. Now, finally, what do we get? Finally, we get 3-PGAL. So friends, we have just seen that under what conditions the Rubisco can function as oxygenase, and it all happens because of the ratio between two gases, that is oxygen and CO2, because oxygen is 21% and CO2 is not even 0.1%. So this is the one reason why this enzyme Rubisco will function as oxygenase. But do you think this is the only reason? No, we have some other reasons as well. For example, if you look at those plants which are growing in arid and semi-arid zones. So I am talking about those plants which are growing in arid as well as the semi-arid conditions, semi-arid conditions. Okay? Now plants which are growing in arid and semi-arid conditions, we know in these conditions the daytime temperatures are very high. For example, if I say Rajasthan, if I take certain part, the southern part of Uttar Pradesh, a part of what is Madhya Pradesh, we know these are arid and semi-arid zones. Now all those plants which are growing in these regions where the daytime temperature is very high. So ultimately what happens? When the daytime temperature is very high, because of the immense sunlight, because बहुत ज़्यादा sunlight आ रही है, so temperature increases immensely. So during this high temperature, we know this high temperature affects the solubility of gases. So if you have a higher temperature over here, if you have a higher daytime temperature over here, we know this high temperature will ultimately, this will ultimately affect the solubility of gases. This will affect the solubility of gases in the aqueous medium, that is water. In the aqueous medium, that is the water. So this is very clear. Now the question is, at higher temperatures, when I am saying that the solubility of gases decreases, that applies to oxygen and CO2 as well. So all those plants which are growing under high daytime temperature, where the temperature touches what is 46, 47 degrees centigrades, ultimately what is happening? Ultimately the solubility of O2 and CO2 decreases in the stroma, the chloroplast stroma, because the chloroplast stroma itself is the aqueous in nature, it is made up of water. But what we know, during this high temperature, even though both the gases, that is CO2 and O2, the solubilities decrease, but in comparison what we see, if we compare these two gases, what we see, that the solubility of CO2, I am talking about the solubility of CO2, the solubility of CO2 decreases much more rapidly in comparison to oxygen. That means the solubility of CO2 in the aqueous medium will decrease. This will decrease much more rapidly at high temperatures. And already we know that the percentage of CO2 is only what is how much? The percentage of CO2 is only 0.034%, and at higher temperature its solubility decreases further. That means the amount of the CO2 present in the aqueous medium, that is the stroma, will further decrease. So what will happen now? What will happen? भाई पहले ही CO2 इतनी कम थी, अब हाई टेम्परेचर पे CO2 की मात्रा जो है, जो CO2 की परसेंटेज है, वो जब और ज़्यादा कम हो जाएगी, what will happen to the enzyme Rubisco? The enzyme Rubisco will start acting as oxygenase. Again, it will start functioning as oxygenase in these plants. Now, this is a problem. This is a problem because all these plants which are growing under high daytime temperatures, they will not be able to fix CO2. फिर उसमें photosynthesis कैसे होगा? Because most of the time we will see that the enzyme Rubisco will keep functioning as oxygenase, and we know whenever it functions as oxygenase, rather than accepting CO2, rather than capturing CO2, it basically captures what is oxygen, and it produces a 5-carbon compound which will be further hydrolyzed to produce a 2-carbon compound and a 3-carbon compound. And we have already seen this 2-carbon compound is toxic. We call it phosphoglycolate, and it can even inhibit the C3 cycle, and the plant can even die. So it remains a problem. What kind of solution is found in these plants which are growing in these zones? Because their daytime temperatures are comparatively always high, and the enzyme Rubisco will always keep functioning as what is oxygenase. It will not function as carboxylase. So again, this is a problem, and how do we overcome this problem? How this problem is overcome by the plants, we will see this later. Look at another condition where this enzyme Rubisco can function as oxygenase. Look at another condition now. The question is, all those plants which are exposed to water stress. If I am talking about a water deficit or water stress, so let us see this water stress. If you look at the water stress, you know, whenever a plant is exposed to water stress, the first defense in the plant, the first reaction of the plant is to close the stomata. We all know this. That means if I say this is a plant, this is a leaf that we have, this is the leaf that we have, and there's a stomata which is present over here, there's a stomata which is present over here. We know the CO2, the atmospheric CO2 will enter through these stomatas. But whenever a plant is exposed to the water stress, what happens is, that is, the stomata would be closed. The stomatas would be closed. And this atmospheric CO2 now cannot enter inside the leaf. It cannot enter inside the leaf because now the plant has to conserve water because it is a water stress, and to conserve water, this will close the stomata, so no transpiration can take place, no water can be lost through the stomata. But once these stomatas are closed, we also know that the gases cannot diffuse. The gases cannot diffuse, and also we know that on the upper surface, look at this, on the upper surface of this leaf, because we have the epidermal cells, and these epidermal cells are coated with cuticle. This cuticle, and this cuticle we know is a waxy substance. The cuticle we know is a waxy substance. So if we close the stomata, we know the CO2 and the other gases, they cannot penetrate these epidermal cells. They cannot penetrate the membrane because we already have a covering of cuticle which is a waxy substance. That means any gas which has to enter inside the leaf, it has to enter inside through what is the stomata only. And if the stomatas are closed during water stress, the CO2 cannot enter, and once the CO2 cannot enter, what kind of circumstances will arise inside the leaf? What kind of circumstances will arise? See this, the circumstances which will arrive is, because we already have a lot of light is already present over here, sunlight is present over here, and when the sunlight is present over here, the light reactions are already going on. By light reactions to ho hi rahe hain yahan pe, and we know light reactions will take place, and when the light reactions are taking place, that means the water is being broken down. We all know that during the light reactions in the plant, the water is broken down and the oxygen is released. The water is broken down and oxygen is liberated. This is what happens during the light reactions, where ATP synthesis and NADPH synthesis takes place. Now the question is, because oxygen is being produced, because water is being photolyzed, now when the oxygen is being produced, this oxygen cannot move out of the leaf because the stomata is closed. Neither this CO2 can enter, ना तो ये CO2 enter कर सकती है, ना ही ये ऑक्सीजन बाहर जा सकती है, और ऑक्सीजन का प्रोडक्शन तो बढ़ता जा रहा है क्योंकि CO2 will keep breaking down, it is being photolyzed. So ultimately what is happening, ultimately lot of this oxygen will start concentrating inside the leaf. Lot of this oxygen will start concentrating inside the leaf in the mesophyll cells, and ultimately we know these mesophyll cells have chloroplast which already have Rubisco. And what will happen? We already know that when the concentration of oxygen increases, the Rubisco will start functioning as oxygenase. This will function as oxygenase. Again, look at this condition. So whether it is high temperature, whether it is the water deficit or the water drought or the water stress conditions, in all these situations, we know the Rubisco, this enzyme Rubisco, will function as oxygenase. This will function as oxygenase. The Rubisco function is oxygenase, and we know whenever this Rubisco functions as oxygenase, this will not capture CO2. In fact, it will interact with oxygen, it will capture oxygen. The 5-carbon compound will combine with oxygen, and we get a molecule of 2 carbon plus a molecule of 3 carbon, and we know this molecule of 2 carbon here is very dangerous. We know this molecule, 2 carbon molecule, is very dangerous. This is what is called as phosphoglycolate. This is highly toxic. So the question arises, how these plants will photosynthesize during high temperatures or during the water stress conditions? All those plants which are growing in desert. Now any plant which is growing in extreme xerophytic conditions or the desert where we don't have water, where we always have water stress, that means their stomatas would be closed during daytime. To any plant which is growing in desert where the daytime temperature can reach what is 48-49 degrees. So how these plants will photosynthesize? क्योंकि अगर वो अपना स्टोमेटा खोलेंगे दिन में तो पानी will move out, the plant will die because of excessive transpiration. और अगर अपना स्टोमेटा बंद करेंगे तो CO2 फिक्स नहीं कर पाएंगे, फिर तो photosynthesis नहीं होगा उसमें. So this is a problem. That means these plants which are growing in different climatic conditions, the plants which are growing in different locations, they have to overcome this defect in the Rubisco, which is the oxygenase. How do they overcome this defect, which is an evolutionary defect in the Rubisco, that it functions as oxygenase? But the question is, we have to find a means, we have to find some way whereby the plant can overcome the losses due to the oxygenase activity of the Rubisco.